All right, can everyone see and hear me okay?
Yes.
Good. Apologies, I've woken up with a bit of a cold today, so my voice sounds a bit more pathetic than usual. But good afternoon, everyone. So I'm Dan from the University of Edinburgh. As mentioned, I'm one of the coordinators of the AQUA-FAANG project. So first and foremost, welcome to this workshop. I hope everybody finds it worthwhile. So I'm going to give a talk to kick things off. I'm going to try to, there we go. Firstly, I'll give quite a brief introduction to the AQUA-FAANG project overall, focusing mainly on the key data sets that have been produced and are being shared through Ensemble. I think that's most relevant to this audience. Afterwards, then I'm going to share some actual results that have been generated during the AQUA-FAANG project for the 2 salmonid species, which had been what my group and collaborators have worked mainly on. I'll spend a bit more time talking about results rather than the introduction to AQUA-FAANG. I mean, many of you know a lot about AQUA-FAANG already, but those who don't, it's been introduced many times, and you can find introductory talks to the project, for example, on the AQUA-FAANG YouTube channel. So just briefly then, AQUA-FAANG is a European project. It's now in the last 6 months of the closing stages. It's one of 6 projects that's grouped together under this EuroFAANG initiative that Garth mentioned, the only EuroFAANG project that's about aquaculture species. And in the project, we've been generating and then analyzing, and relevant to today, sharing functional annotation data for the 6 key aquaculture species that are shown on this tree on the left. I'll just get a laser pointer. Yeah. So these are the major farmed fish species in Europe that make up about 90% of all the production by value. And my talk today is going to focus on the salmonid species, Atlantic salmon and rainbow trout. These split off about 25 or 30 million years ago. They are characterized, or their genomes characterized by a shared ancestral whole genome duplication event, which is of a lot of scientific interest, including to researchers in AQUA-FAANG, and I'll come back to that soon. This slide gives a bit of an overview of the key data that's being generated and shared in the AQUA-FAANG project. So I'll focus on the 3 main data sets of functional annotation maps that have been produced for each of the 6 species, which was done in as standardized a way as is possible. So the project has generated more data than what I'm presenting or summarizing here, but in terms of what will be shared through Ensemble, this is really the key outputs of the project. Excuse me. So these 3 functional annotation maps are aiming to capture the dynamics of genome activity across embryonic development. We call that dev maps, in adult tissues from both sexes, in both mature and immature stages of development. That's called body maps. And after immunological challenge, which is called immuno maps. I'll come back to those in a bit more detail when discussing the salmonid data. So the different types of functional genomic data generated are summarized on this figure on the right, which was made by Paran, a postdoc in my group. So we've generated ATAC-seq data, which allows us to identify open chromatin regions that are accessible for transcription, and then ChIP-seq data allowing histone modifications to be quantified. And in AQUA-FAANG, we've focused on 4 histone modifications, which together allow us to distinguish promoter and enhancer regions in different states. So for example, open chromatin by H3K27 acetylation and H3K4ME1 with low H3K4ME3 can identify an active enhancer, whereas other marks can identify other states like H3K27ME3 is laid down by the polycomb complex and marked genomic regions that are where transcription is being repressed. And we're also measuring the actual activity of coding genes and small RNA genes using RNA sequencing. So AQUA-FAANG is filling a big gap in knowledge in terms of how the genomes of these different fishes are being controlled under different biological conditions across life. In addition to sort of helping us understand the biology and adaptations of these different fishes, the functional annotations have more practical uses, especially to ongoing attempts to predict trait variation using genetic data, particularly for selective breeding programs such as the ones that Aquagen run, that Tim is obviously working with. And the idea here is that obviously many traits have a genetic component, for example, disease resistance in the example shown here, but we rarely know which genetic variants are actually causally involved. This is a big challenge because we have such an enormous number of genetic markers that are in linkage to equilibrium with the causal variants, and we have little way of knowing, especially for variants in non-coding regions, which are the most common ones, what they're actually doing. And the idea in theory is that functional annotation can help us prioritize and identify causal genetic variants, particularly where it's most hard to do so in non-coding regions, identifying variants that overlap with regulatory regions that are activated or repressed under different biological conditions, or in animals that might have different traits. And this information may improve the accuracy of precision breeding methods, though this idea is still very much in an evolution and is something being investigated within AQUA-FAANG ideally. So as the main subject of this talk, I'll just quickly introduce salmonids. Salmonids are obviously a really important group of fishes, including for global aquaculture. Something like 2.5 million tons of Atlantic salmons produced each year, and another approximate 1 million tons of rainbow trout around the world. But also wild populations have great conservation value and cultural value to humans, and they're widely used in scientific research for a large number of purposes. Excuse me, as I mentioned earlier, salmonids also have really interesting genomes due to a whole genome duplication event that occurred in the ancestor to this whole family. This was an additional genome duplication to the one that occurred in the teleost fish ancestor and the events that occurred very early in vertebrate evolution. And this whole genome duplication has left the genome in a complex state where there's global retention of duplicated regions and genes and regulatory elements that show varying levels of sequence divergence due to the way that the genome evolved after it was duplicated through evolution. So salmonids are an important vertebrate model system to understand the evolutionary impact of these genome duplication events, which is of wide interest, especially on genome function and regulation. And that's an area where there's very little, it's very underexplored, and AQUA-FAANG can make sort of great leaps forward with our data. So, excuse me, the AQUA-FAANG researchers working on salmonid species have sort of overall got the following aims. The first is to understand how the genome's being regulated from embryo to adult using the DevMap and BodyMap data, and that's being done in a comparative framework, considering both the salmonid species and providing insights into genome evolution. The second major goal is to understand how the genome is being regulated following immunological challenge to pathogen signals. And I'm going to come back to this very briefly at the end of the talk. And another important objective that I won't cover really today is to take all the functional annotation information that we produce and overlay it with existing or new panels of genetic variants. So that could be SNP markers, but it could also be structural variants, which have been studied in both species. And this has a number of downstream applications, for example, in selective breeding. And obviously that side of things is going to be covered by a talk and one of the ensemble tools, VEP, and I think that will be covered tomorrow, which is great. So moving on to talk about some of the analyses. This slide lists all the individuals that have been involved in analysing the DevMap and BodyMap data in the two salmonids. And this group's currently working towards sort of a large flagship paper that we hope to submit at the end of the year. So you can see this involves institutes from the UK, Norway, and France, which have together as a group generated and or analysed all the data to be presented in a moment, all supported by building tools that we've used in comparative analyses. I won't name all the individuals involved, but I'm presenting data on behalf of all of them, and I will give credit for figures and images and things as I go along. So the DevMap samples, excuse me, capture 14 stages of embryonic development that are matched across the two species for RNA sequencing. So this is from fertilization onwards, including the cleavage stages, then the blastulation, when the genome is activated, gastrulation, when a lot of important patterning happens, segmentation and somatogenesis, leading up to organ development. So this is really similar resolution to studies that have been done before in model species like zebrafish, but it's a much more challenging sampling effort in salmonids, as it takes about 5 weeks to get to this same stage of development in these species. And in AQUA-FAANG, we have 6 key embryonic stages that we've attempted to standardize across all the 6 species. And for these samples, in addition to RNA Seq, we have the CHIP data with 4 histone marks in the TAG-seq. And for the body map data, we have a standardized panel of tissues as listed here in both sexually mature and immature stages, and where we've generated RNA attack and CHIP-seq with the 4 histone marks shown on the left. And an important thing about AQUA-FAANG is that the samples are closely matched across the species, which makes for a really unique data set for comparative analysis. So we've generated in each species in the region of 1,000 sequencing libraries, across both species in the region of 1,000 sequencing libraries. So really a very large data set compared to anything that's been done previously in salmonids. All the data and associated metadata for these samples was submitted to the FAANG data portal. So it's publicly available in its sort of raw format with lots of information needed to help support reproducible use of the data. Just a quick note on data analysis. So the team involved have performed data analysis for RNA attack and CHIP-seq data sets using NF core pipelines, and the data has been separately uptaken by Ensemble to update gene annotations and produce new regulatory annotations, which both will be introduced and explored heavily during this workshop. I've given some information just about the actual analysis parameters that were used. I also won't talk about small RNAs today, but a separate analysis is well underway and at a mature stage, and that's been done by a team that runs the fish microRNA database, which is coordinated within AQUA-FAANG by INRA, one of the partners in France. And that group is producing a novel microRNA annotation for all 6 AQUA-FAANG species using the small RNA libraries, and that will be available later this year publicly for those who are interested. I don't plan to say a huge amount about the primary data that we generated, as I want to focus more on findings. So you'll just sort of have to take my word for it that we've generally been happy with the quality of the data that's been generated for both the salmonid species. Obviously, we've lost a few samples along the way, and not everything's been all rosy, but that's the way of it. But generally, the data has been of high quality. A couple of quick illustrations. These are just some PCAs for the RNA Seq expression level values from the adult tissues in both species. At both stages of maturity, you can see that there's nice groupings by tissue type, and a mirrored pattern across the two species, which gives a lot of assurance in the quality of the data. This slide shows PCAs for the peaks that were derived from the CHIP-seq in the embryos in both species, which generally show a good clustering of replicates by stages. We get a good number of peaks across the different sample types in the project as well. So now I'll move on and actually share some findings, some results, and I'm going to start with gene expression results. On this slide, this is data from Atlantic salmon embryogenesis, so the DevMap data. So we've used a method quite a lot in our research called self-organizing maps, which is an unsupervised artificial neural network clustering method, where the clusters or the classes retain an order such that the more proximal clusters share greater similarities. That's the idea, and we found this has worked really well on the data sets we've produced. So the heat map shown here on the left part is from a clustering of 14 embryonic stages, where you can see, for example, clusters or classes representing maternal messenger RNAs like class 5, genes that are being upregulated specifically in early embryonic stages, such as the blastula, probably linked to zygotic genome activation and during gastrulation, as well as genes that are upregulated in late embryogenesis, probably involved in organ development, among others. If you look at part C, Diego, who did this analysis, has successfully employed UMAP, dimensionality reduction method, to visualize in a summary sort of way the trajectory of gene expression from the early to late embryo stages. So here, each dot on that UMAP is an individual gene, and we can visually look at the expression of the entire transcriptome through development. And the colors there are linked to the colors that are on this SOM clustering, going from early to late development. Excuse me. And a full SOM clustering analysis is shown then on the right, and Diego has done this approach separately for both species, and you can see common clusters for both species, like these examples I've shown in blue for rainbow trout, and that provides us with a really useful basis for the planned comparative analyses. It also gives us confidence that we've matched across the stages appropriately between the two species. And this allows us, for example, to compare the genes found in common clusters across the species and see how many of them are orthologous genes, which would be our primary expectation. Or we can also look at the duplicated genes that have been retained from the whole genome duplication event to see if they've maintained or changed their expression in evolution. Here I'm showing independent performed UMAPs by Diego for both the different species, where you can see we observe a very similar trajectory of embryonic transcriptome development in both the species on the left here. And for comparative analysis on the right here, Diego has performed a SOM clustering analysis, integrating the data from both species in a single effort, which again we're using to infer cross-species conservation or divergence in gene expression at different stages of development. And on the right here, you can actually see a clustering analysis where both species are included. So you can see the different clusters contain blue and red, which shows the contribution of genes coming from the different species. So this slide then shows a busier SOM analysis for the body map data for Atlantic salmon. And we have a very similar map for rainbow trout, which nicely organizes the data set into modules of gene expression across tissues and stages and sexes. I won't go into great detail, but just to highlight a couple of examples to illustrate the value of this method, we can identify genes that are upregulated in particular tissue types in both sexes at both stages of maturity. So these are just, for example, signature genes in the gill tissue. Or we can use this method to identify more stage-specific expression in particular tissues. Like here, we're looking at stage-specific gene expression in the testes, depending on whether the fish are sexually mature or immature. And so on, you get the picture, I suspect. Diego's also trialed the use of UMAP dimensionality reduction as an alternative way to visualize this data. This is this artistic paint blob on the left. Here, the colors on the UMAP match with the different SOM classes on the right, and the center of the UMAP then is capturing genes that have more constitutive expression across different tissues and stages of life. But we can also use the UMAP to visualize other SOM clusters, which are showing much more specific expression, for example, in these different tissue types. So these sort of global approaches offer a real useful way for us to explore the gene expression in a global sense and link that back to regulatory elements. And I'm going to move on and talk about some of the data we've generated on gene regulation now. So you may be familiar with AQUA-FAANG, which is a tool that allows us to predict chromatin state across the genome using the ATAC and ChIP-seq data, which is informed particularly by the different histone marks. So the research group has performed this approach for both salmonid species separately for the DevMap and BodyMap, in each case integrating all the ATAC and ChIP-seq data. And there's example state predictions shown in this figure here. So Chrome-HMM and I think a related tool Segway are used by Ensemble in their regulatory annotation pipeline. I think Garth's going to cover that tomorrow, so I'm not going to try and explain that properly, except to say that these methods effectively break the genome up into segments that represent different chromatin states, which we then need to interpret based on our knowledge of the underlying features. So, for example, we can identify enhancer regions based on the combination of open chromatin and the presence of H3K27 acetylation and or H3K4ME1 to the exclusion of H3K4ME3. And there are many other classes too. For example, you can see repressed polycomb is marked specifically by H3K27ME3. And we can also identify regions of open chromatin that lack any histone marks, which we know are highly enriched in CTCF binding sites. And these have previously been identified in zebrafish, so are an interesting category of element in their own right. Also, just to note how we used ATAC peaks to define high confidence regulatory elements. So these high confidence ATAC peaks were identified using the IDR method, which was done by ENCODE. And then we take those ATAC peaks, which are effectively the ones that show highest reproducibility between the biological replicates used. We take those and we annotate them with the Chrome-HMM predictions, effectively following a method that was done very successfully in a paper that was published recently by the zebrafish functional annotation community. That was, first author on that paper was Demir, who's also working heavily with us on AQUA-FAANG. So we're very happy with this method, but any results I present on regulatory elements shown from here on were defined this way. You can see examples of the number of open chromatin regions defined by this method in this table for some of the sample types, and on the graph on the right for embryo stages, where you can see the genome's becoming increasingly permissive to transcription or more open as development proceeds, which is expected from previous work in zebrafish. We've also assessed transcription factor binding motif enrichment in open chromatin regions that are defined as active promoters and enhancers based on the Chrome HMN data. Here I'm showing some data that shows evidence of enriched transcription factor binding motifs in active enhancer elements for both the body map and dev map samples in separate heat maps, so the body map on the left and the dev map on the right. So this gives us really assuring results in terms of data quality. So for example, in muscle, we see enrichment of key known muscle transcription factors like SIX2 and MEF2. In brain, we see enrichment of neural transcription factors like neuro-D. In embryos, we see enrichment of HOX genes, which is very much HOX transcription factors, which is very much expected. In gonads, we see DMRT3 for testes, which is again expected. And in the liver, we see things like hepatic nuclear factors, FOXP1. These are master transcription factors for liver and hepatocyte function. So the data is looking all good in that sense. The data derived purely from embryos on the right also gave us expected transcription factor motif enrichments across the different embryonic stages, but I'll maybe not cover that just in the interest of time, which is, I assume, ticking away quickly. So focusing now on some regulatory data that was generated for, this is Atlantic salmon embryo data. This Venn diagram on the left is showing the overlap of stage-specific attack peaks in the Atlantic salmon embryo. About a quarter of all the peaks are common to all the stages in the embryo. They mainly overlap with promoters and likely largely represent things like housekeeping genes. You can see the greatest number of stage-specific attack peaks are activated in late embryonic development. It's quite clear that the chromatin accessibility is very dynamic. So to better sort of categorize this, the dynamic of chromatin accessibility, Demir has again used SOM clustering, taking a union of all the attack peaks in the salmon embryo. And we have a heat map on the right here that shows the most dynamic classes of open chromatin regions. You can see we can readily identify open chromatin regions that are separating different stages in the embryo. For example, the late blastular stage, when the genome's being activated, or peaks that are specific to the late embryo, which are likely involved in things like organogenesis. So again, you can see the power of this, these sorts of global clustering methods. And taking a similar SOM clustering approach with all the high confidence attack peaks or regulatory elements defined across all samples generated for the dev map and body maps, it's possible then to identify signature open chromatin regions or regulatory elements for different stages of development for different tissues, which Demir has summarized on a UMAP plot where every single dot is an attack seek peak, as well as in the accompanying heat map on the right here, where you can see we can readily identify peaks that are defining liver or muscle. But you can also see the peaks in the embryo and in the brain, and you can also see stage-specific and sex-specific effects within this data too. So again, really interesting data, I would say. I mentioned the genome duplication quite a lot. This is of a lot of scientific interest to my group and collaborators, and I'd like to highlight a bit of data where we've tried to understand a bit about the evolution of gene expression and regulation linked to this genome duplication event. And I'll start with some gene expression data before moving on to regulation. So this plot shows an effort to compare the different sample types that we have in terms of the extent to which the duplicated genes retained from the genome duplication event have changed in expression, or you can also look at it in how conserved they are in expression, using this method called the Jensen-Shannon distance, which effectively summarizes the similarity between a global distribution of TPM differences between pairs of duplicated genes from the whole genome duplication event. This data is again for Atlantic salmon, but the results look really similar for rainbow trout. And here on this plot, we're effectively ranking how much per each sample type the duplicated gene pairs have diverged or are conserved in expression. You can see then that in embryo stages, the duplicated gene expression is least conserved in early stages, like in the maternal messenger RNA, and leading up to when the genome is being activated, and highest constraint on the duplicate gene expression, or the most conserved duplicate gene expression, is in the late embryo stages. Whereas you can see tissues in the adult stages also show a very high degree of variation in the extent to which the duplicated genes have diverged in expression, with liver and gonad, particularly the testes and the ovary in both stages, showing the highest degree of expression divergence among the duplicated genes. So the graph here on the left is the same data, but what Diego has done is more simply averaged the divergence of duplicate genes across all adult tissues for both maturity stages. The results are consistent with the so-called hourglass model of development, which has been long posited, where evolutionary constraint on phenotype is meant to be highest in late embryonic development in terms of the whole life, and the lowest in very early embryonic development and later stages for ontogeny. That model's been supported comparing orthologous gene expression across different species in several different studies, including the study, this quite famous Amphioxus genome paper by Martinez et al., where you can see a similar pattern. But as far as we know, it hasn't been described before for duplicate gene expression from whole genome duplication event. So that's quite a new finding. Excuse me. So we've also been looking into the evolution of duplicated regulatory elements. This is the most latest data that we've generated, and it's quite a fast-moving analysis. But to study duplicated regulatory elements, we first had to build a genome alignment that included both the salmonid species that captures both the duplicated regions retained from the whole genome duplication event, alongside an outgroup species where the genome is closely related but didn't undergo the whole genome duplication event, and we used this species northern pike for that. So in effect, this genome alignment resource allows us to compare the conservation of sequence in the genome retained from the whole genome duplication event in both the salmonid species in relation to the presence of regulatory elements or anything epigenomic that we've defined in our ATAC and ChIP-seq data. I've tried to summarize this in this figure at the bottom left. So here we have duplicated regions A and B that have been retained from the whole genome duplication event, and they can be conserved in both salmonid species, such as at the left side here of this plot, or we can have a situation where just one of the duplicate is conserved in both of the salmonid species because it was lost in the ancestor to salmon and trout after the genome duplication. Or we can have sequence that's private to one chromosome in one species, which could potentially have been acquired later during evolution. So some of this might be, for example, repeat sequences that accumulated during evolution. Then onto that framework of sequence level conservation, we can layer the actual observed data that we see for the ATAC-seq, and we can categorize it. So for example, we find that somewhere around 11% of all Atlantic salmon attack peaks are conserved between both duplicated regions in both salmonid species. A similar number, about 8% of all the Atlantic salmon attack peaks, are present in only one of the duplicated copies in both species, despite the fact that the other duplicated sequence is conserved. So suggesting that across all the different sample types we've looked at, we can never find open chromatin region in this other conserved region. That doesn't mean it's not active in some other type of sample that we haven't captured in our data. And a much larger number of the attack peaks, almost 1/5 of the Atlantic salmon attack peaks, are private to one chromosome in Atlantic salmon, even when the sequence is conserved across both duplicates in both salmonid species. And so on. We can identify peaks that are conserved in all different possible combinations. And the take-home message is really that we're able to successfully classify and describe the evolutionary history of regulatory elements during salmonid evolution and link that back to the ancestral condition in terms of sequence conservation, at least. But this work is at an early or fairly early stage now. Here we're summarizing attack data across all sample types, but we can get a lot more scientific insight if we look at the results in finer details across the different sample types. If we look at things like transcription factor binding motifs and how they've been split between duplicated elements to alter gene expression during evolution. And these are all things that we very much have planned for the near future. But I wanted to come back to the analysis I presented in terms of Diego's work, looking at the conservation of duplicated gene expression across the different samples. Manu, who's the guy who's been working on the regulatory elements, has recently attempted to perform a matched analysis using the regulatory element data. So this plot from the right shows two major categories of regulatory element activity for regions where the duplicated sequence is either retained and you have two peaks, or where the sequence is unique to one chromosome and there's an attack peak there. And what you can see is that where you have attack peaks in both duplicates with conserved sequence, the highest conservation, if you like, of duplicated regulatory elements, this is occurring in the late embryo stages, and it's lowest at the early embryo stages. And looking at it reciprocally, where you have attack peaks that are unique to one chromosome, you have the lowest level in the late embryo and the highest level in the early embryo. And overall, you can see there's quite a strong coupling between the duplicated regulatory elements and gene expression. So we see an overall very similar pattern. This is a broad analysis that's still in progress, and we're going to look at this in more detail, splitting the attack peaks into different categories of regulatory elements like promoters or enhancers. And we basically have a huge amount left to do in that space, but the results are looking really interesting. So I'd like to finish up just by briefly presenting some results on the third functional annotation map, the salmonid immunomap analyses. This is work that's been led by the University of Aberdeen for Atlantic salmon and INRA for rainbow trout. And these two teams have worked very closely together, including on downstream cross-species analyses. My group's collaborating with both teams on that effort too. There's already a paper that's been submitted, and there's others in preparation. And in short, and in common with the other AQUA-FAANG species, the salmon and trout were stimulated both in vivo by injection and in vitro in primary immune cell cultures using either poly-IC, which is a mimic of viral double-stranded RNA that activates the interferon or antiviral response, and or with inactivated Vibrio bacteria, which activates an inflammatory response to the bacterial signals. I was just going to very quickly present a tiny portion of this data for Atlantic salmon, which was done by Shamir, who's at this meeting in Sam's group in Aberdeen. And I think this is really interesting new data. So the data I'm showing here is Shamir's data just for poly-IC. So comparing fish that had been injected with this viral mimic to controls 24 hours after injection. And the data was produced and analyzed in a very similar way to what was presented before for dev maps and body maps using NF core pipelines. Shamir's also performed an initial method to look at differential chromatin accessibility with the ATAC-seq data, or differential histone modification with the H3K27 acetylation. So the latter representing active regulatory elements, so it could be the promoter or enhancer elements. So you can see the elements on these graphs that are upregulated or downregulated on these volcano plots, in response to the poly-IC. And on the right, you can see the gene ontology terms for genes that are in proximity to these differentially regulated elements. So for the up category, strongly reflected are a range of antiviral and immune response terms, including terms related to interferon. And I think these regulatory elements overlap more than you'd expect by chance with genes that show differential expression in response to poly-IC. So overall, really nice results that validate that the genome really has been activated in regulatory terms in response to these to poly-IC. And finally, just to illustrate some nice visualizations that Shamir shared with me recently of specific genes that showed increased accessibility upon poly-IC stimulation. This included many genes that are known to be involved in the antiviral response. So here we're looking at known interferon response genes that showed upregulation, like expression upregulation to poly-IC, but that was coupled to activation of proximal regulatory elements that contain interferon sensitive response elements, which I think the interferon complex binds to. So overall, this is validating really the quality of this immunomap data set in Atlantic salmon, and I'll be really excited to see more results that come out of this work in due course. And I don't have any grand conclusion slides. That's really all I wanted to say today. I've probably used more than my allocated time, and I'd happily answer any questions if there are any, and I hope everyone